Yeah, so there've been a last minute change. Caroline was not feeling well and couldn't give the talk today, but her excellent student Adit Radhakrishnan will give the talk. So please, Adit, go ahead.
Yeah, no, thank you for thanks for the introduction, Misha. I have to also say I've been working throughout my whole PhD with Caroline and Misha as well, so I really consider him one of my code-wisers too. But the topic of the talk actually hasn't changed too much. I'll still be discussing over-parameterized autoencoders and applications of these in genomics, again, highlighting the benefits of over-parameterization overall. And I know everyone here is probably familiar with benefits already from double descent, but I think there's been a lot of extensive work now understanding the generalization benefits of over-parameterized models in regression and classification. And actually, Lorenzo is a great introduction or a great segue to this, but there hasn't been too much work studying over-parameterization in unsupervised learning, and in particular in representation learning. And so this is the sort of question I wanted to focus on for the talk today. And I wanted to discuss probably the most fundamental model in unsupervised representation learning, which is the autoencoder. And so what is an autoencoder? Well, I just think about it as a neural network that's taking some input data that's unlabeled, and I want to learn a representation for this data by feeding it through a neural network and asking the neural network to just reconstruct the original images that I feed in. And the typical way an autoencoder is motivated is that these models, of course, like one really simple map that can do this, that can implement an autoencoder, is the identity function. But you don't want the model to learn an identity function. This is like a useless representation for your data. So the idea is that autoencoders are typically bottlenecked. You typically have some latent space, and I've drawn it down here in a schematic, but you want to prevent the model from learning the identity function in some way. And so I think autoencoders and over-parameterization have been mixed for many, many years because of this one fact. Like, they can learn the identity function if you make the bottleneck large enough. But the question is, even though they can do this, what do they actually learn when you have this, when you train them with gradient descent? Okay. So to kind of showcase a very strange but interesting phenomenon, we just took the basic example of an over-parameterized autoencoder. You can take like a one hidden layer neural network. And in the middle, instead of having that bottleneck, you just completely eliminate the bottleneck. And you just train on one image. So this model, all we're doing is we're feeding in one image of these two rabbits and asking it to reconstruct the image of the two rabbits using mean squared error. And of course, it can perfectly do this. This is like one sample. It's way over-parameterized. But the question is, what happens when you feed in test data? So if the autoencoder, even though it can, if it learned the identity function, this would be useless. Any image I feed in, I'll just get that image back. But what actually happens is that no matter what image you feed in, I mean, basically almost everywhere, you'll get exactly this picture of the two rabbits out. So you can feed in random noise, you can feed in realistic looking images, you can feed in like shapes, things like that, you'll get this out. And I always get this question, which is, maybe I'm cheating in some way. Maybe the network has a biased layer and I've just encoded the image in that biased layer. This network actually has no bias layers. If you feed in this image that's all zeros, you will actually get the all zeros image out. So pretty much everywhere else, though, you'll get this picture of two rabbits. So it's learned a very interesting function that's constant almost everywhere, or near constant almost everywhere. And so, of course, this is only with one example, but already this is a bit surprising. And the reason is that, well, first of all, like I said, there's infinitely many solutions in this over-parameterized setting. And one of them is this identity map. And actually, certain linear neural networks will learn this identity map. And it is provable that shallow linear convolutional networks, like one layer, will actually do exactly this. You'll feed in an input, you'll get that input back. It'll just spit it back in. So something really interesting is happening here with this nonlinear network. And in fact, even if you have like one example or multiple examples with a linear fully connected network, this is just like regression. You'll get this minimum norm solution, this minimum L2 norm solution. And this is just a projection onto your samples. So any input I feed in, I'll either get this picture back or I'll get a constant multiple of the picture back. And you can get some weird mixtures. And in the case of 2 samples, you'll get some very weird mixtures of like a dog and a plane. And that's kind of what's being shown here. So it's essentially a nonlinear phenomenon that's happening, and we can't really understand it with linear neural networks. But of course, like this is all for one sample. A natural question is, what happens when you actually train an autoencoder on many samples that you want to learn this representation? So after training on 500 samples for ImageNet64, what we did is we did the same experiment again. We took some images, we corrupted them in some way, and then we just fed them into the autoencoder and looked at what the reconstruction was. And it turns out the reconstruction looks like a noisy version of this sample from this training set that's uncorrupted. But the best part about an autoencoder is that it's a map from one space to itself. So whatever output that I get, this is also an image, I can just keep feeding that back in to the autoencoder and see what happens. And so if I keep doing that, you'll just see very soon that, I think like within 6 steps, you just perfectly reconstruct or recover the training sample that you had fed in. And so this is kind of like a nice generalization of the property that we had for one image, but now just for many, many images. And of course, we did this more systematically across many different types of noise, many different types of corruptions. And you can really see here that the autoencoder is able to recover these training examples. So for those of you who are familiar with the neuroscience community, this is a particular example of associative memory. Given some corrupted sample, I'm able to associate it with a sample in my training set and recover. Of course, this is a cool empirical phenomenon, but we really wanted to understand what was happening theoretically. And there's some nice math that essentially we're dealing with the autoencoder, an iteration of this autoencoder as some dynamical system. And so what this work is really showing from a theory perspective is if I'm looking at an over-parameterized system where I'm just learning points that lie on this identity in one dimension, these are my training samples. Of course, there's many solutions in this over-parameterized setting that perfectly fit the training data, but gradient descent is not recovering one of these crazy solutions. This would be like a case of overfitting in some way. But what it's actually doing is it's learning a function that's contractive around the training examples. And what I mean by that is just, if I was to think about this function around each training example, it just looks kind of flat. It's locally flat. And so the best part about this is that when you iterate a function that stores these training examples, it's contractive around the training examples, you exactly get a piecewise constant function. And that's exactly the phenomenon we were seeing on that slide with iteration. You've learned your model that's contractive around the samples. And so if you take a corrupted sample, which is like nearby in some space, and I just iterate the function on that, it's converging to this flat region. And so you're able to perfectly reconstruct these training examples. And mathematically, the 2 conditions that you kind of need for this to happen, the first one is nicely already encoded in this autoencoder objective, which is that every training sample has to be a fixed point, which means that you're perfectly reconstructing your training examples. The second condition is the tricky one, which is non-obvious why gradient descent is giving us this for free. But the idea is that if I look at the Jacobian of my autoencoder, or basically the derivative, right, around each training sample, if the eigenvalue of that Jacobian is less than 1, or in this one-dimensional case, the derivative is less than 1, then these fixed points become attractors. So when I iterate the function, I exactly recover the training sample. These are the 2 conditions that you need. So again, the first condition is fixed point is easy. This is exactly what you get from interpolating data. The second one is the non-obvious one. Why is this happening? But just to see that you can actually get some very cool dynamics from these autoencoders. Unfortunately, in high dimensions, we can't have these nice visualizations in some way. But in 2 dimensions, if I just take these 6 points in 2 dimensions, which are my stars, and I train an autoencoder to perfectly fit them, I can then just look at the whole space and iterate all the points in the space and see which training example this model converges to. And so already you can see very clearly that the Basins of attraction, which is these different colored regions, so here this entire red region would converge to the red star. you can see that these are very different from what you would get with something like one nearest neighbor. And typically you would get like these Voronoi diagrams, but this is not the case with autoencoders. You can get some very strange and interesting Basins. And I should say, it looks here like all the Basins are connected in some way. You have these different connected components, but actually, we have some work now that shows that you can have autoencoders that store attractors with disconnected Basins of attraction. So still a lot of interesting geometry that comes out of this. And for those of you who are interested in visualizing, like, how does the flow actually go on this space, just as a simulation, if I just drop some number of points shown in black, and I feed them through this autoencoder over and over again, you'll see that they just converge pretty nicely. And in some cases, you can see that like this green point is straggling a bit, but over time it will converge to this one training sample. So you can always recover the training examples in this way, and there doesn't seem to be any other fixed point attractors in this space. Great. So that was in 2 dimensions, but we wanted to analyze what do these Basins kind of look like in higher dimensions. And so we just ran our model on many, many different corrupted samples and just tried to check the recovery rate. So if I take some picture of the rabbits and I apply this noise template to it, do I actually recover the rabbit? And it turns out in most cases, this model is pretty robust to noise, but this is a fully connected neural network. And as you imagine, in higher dimensions, you do get something very similar to a one nearest neighbor metric for how attractors attract different samples, but it's not exactly the same. And so understanding this metric for the Basins of attraction, this is still a big open question for us. Okay. So just to show a cool example, this attractor phenomenon you can get just from autoencoding, but it turns out if you have other types of data, like sequences or videos, you can actually recover these as well in a very similar way. And the objective is almost exactly the same as autoencoding with a slight difference. So if I was just to think about a list of digits from MNIST as a sequence instead of a series of examples, in autoencoding, what I would typically do is if I have 6 digits, I just take each one and map it to itself. That's the autoencoding objective. So these are all fixed points. But on the other hand, if I was to not do that, if I was to take each digit and map it to the next digit in the sequence, and then have that last digit wrap around, this would be sequence encoding. And at first, it might seem like this would break this fixed point property of autoencoders, where if you want to store an example as an attractor, you would need it to be first a fixed point, so it maps to itself. But actually what's interesting is that if you train a sequence encoder like this, you'll see that it's actually much easier to recover the whole sequence via iteration than to recover any individual example from autoencoding. And just to give some intuition as to why this is before I show the examples, one reason is that if I just look at a function, f, which maps to 0 to 1, 1 to 2, and so on, up to 5 to 0, if I just look at f composed with itself 6 times, that composition of functions maps 0 to 0 and 1 to 1, and so on. So that function has stored now your whole sequence effectively as an attractor, as a fixed point attractor. But I'll go over this a bit more carefully in a few slides. Okay. So just to give a fun example of sequence encoding, of course, one classical example is you can take any movie. This is just a sequence of frames, and I can do exactly what I did on the previous slide. Instead of taking each image to the next one, I just take each image of this movie into the next frame and then wrap it back around so the last frame maps to the first. And so I did this with around 400 frames of a Mickey Mouse film, Steamboat Willie. And then what I do is after training this neural network, this sequence encoder, I just feed in completely random noise, and I just iterate this model on this random noise, and I see what I get. And if I just play this video, you'll see like almost immediately, it's just perfectly reconstructed the film and you recover this. So, and you'll see eventually it starts looping on itself. But this is of course just one very long sequence. It just immediately recovers it and starts looping. If you do this with multiple sequences, you can actually get similar behavior. Oops. And so this is just, I took 2 sequences of digits on MNIST, if the videos want to play for me. And I just, one sequence is just digits from 0 to 9 counting up. The other is 9 to 0 counting down. And I just trained the autoencoder on both sequences, or sequence encoder on both sequences simultaneously. And you'll see like very clearly after iterating on some noise, I can just get one sequence which is counting up, the other sequence is just counting down. And so this attractor phenomenon seems pretty general as long as you have some sort of fixed points in your objective. So gradient descent is really doing something interesting in this setting. But of course, we can again do all of the same visualizations for what this looks like in 2 dimensions. And in 2 dimensions, before we had attractors where we had each individual star as a fixed point, but now we have these orbits. We have these groups of samples which form little discrete limit cycles in some way. So when I start iterating a function, when I start iterating this function on any sample in this space, you'll see that it just starts falling into this loop. And so that's exactly what I've visualized here. I take some points, I drop it in this space, and you'll see very quickly it starts looping around. And you'll see like, you know, I didn't believe it at first because some of these were taking a while, but if you do wait long enough, you will get that all the points converge into these orbits. So it's quite nice, I think, in this setting that you get this behavior. Oops. So the question then is, now that we've, you know, very robustly see this, you know, this pattern in practice, how can we actually prove something like this? And can we actually do things like prove sequence encoders maybe are more efficient at storing examples as attractors than autoencoders? And so at the time of writing of our paper, we had a nice theory for one sample. And the idea is that if you have just a neural network that's trained exactly on one sample, it can be arbitrary depth and smooth nonlinearity, since phi is a smooth function. But you just have one sample. So I have an xi that maps to some yi. We can actually just recover a closed form for what this solution looks like under a gradient flow. And there's some nice invariances that we can come up with. If you're familiar with the papers by Sanjeev Arora's group, this is akin to that alignment or balancedness phenomenon for linear neural networks, but this is for a nonlinear setting. And so once you have a closed form solution for one example, we can just compute everything we need to see if autoencoders store one training example as an attractor. We already know that it'll store it as a fixed point. So we just need to compute the derivative or Jacobian at this fixed point and just compute the top eigenvalue. And that's exactly what we did. And we can actually prove that basically for any nonlinearity that's smooth, there exists some initialization that will give you this example stored as an attractor. I think what's pretty nice is that this same analysis can be kind of cleanly extended to a sequence encoding setting. It's not completely faithful, but one way to think about sequence encoding and compared to autoencoding is I can actually look at if I have n samples or n frames in a sequence, what I could do to learn all of these samples as fixed point attractors is I can just take n samples and solve my, you know, one hidden layer or whatever number of hidden layers neural network, but take n different neural networks to do this. And for each one, I need this condition that the top eigenvalue of this Jacobian at these samples is less than 1. And so this is pretty painful. If I have n samples, somehow each eigenvalue has to be less than 1. That's a very stringent condition. But what's nice is that if I do the same analysis, but instead of doing each example to itself, I actually take each example to the next example in the sequence and loop it around. I can still solve these using the same theory from the previous slide. I can just compute what the solution is. But then what I can do is, if I want to see if the whole sequence is stored as an attractor, I just take the composition, these n functions, and then I look at the top eigenvalue of the Jacobian of this composition. And what actually is interesting that comes out is that the condition that the whole sequence is an attractor, or is a discrete limit cycle, is that the product of the eigenvalues has to be less than 1. So in the top case, just to summarize this again, if you have n networks, you somehow need n autoencoders, you need each autoencoder to have eigenvalue less than 1. But for the sequence, you just need that the product of these is less than 1. And so having that the product of a set of numbers is less than 1 is a much less stringent condition, and actually gives some intuition that sequence encoding is more efficient. So if you do need to store data as a limit cycle and be able to recover it, sequence encoding is the way to go. And so in practice, actually, we did this. We wanted to showcase a nice example of this. And there's a lot of numbers here, but I can summarize the main point. In figure A, what we did is exactly this example on the previous slide. We took an autoencoder just on 100 MNIST samples, and we just vary the width and the depth of these autoencoders and just train. These are all fully connected. And we just train these and see how many of those examples can we recover? How many of those have eigenvalue less than 1 for the Jacobian? And you can see here, like, it's very painful. It's if you do 31 layers deep and then 512 width, you still don't recover all the examples. But what you could do is, if you wanted to recover them, you can start grouping your examples into mini sequences. So you can have 20 sequences of length 5, 10 of length 10, and so on. And if you do that, you can very quickly see that even with 10 sequences of length 10, you can recover all the sequences with a depth 6 network and a width 512 network. And in the best case, you can actually recover all of the samples if you store them as one big sequence of length 100. You can recover all of them with a depth 1 network and a width 512 network. So this is kind of nice that you get this very clean benefit to sequence encoding for associative network. Great. So this is essentially what I wanted to say about the inductive biases of over-parameterized autoencoders. So we haven't said anything about, are these actually good for generalization or anything like that? But what we do know is that over-parameterized autoencoders, when you train them using standard optimization methods, they learn maps that are contractive around the training samples. So these are maps that are flat around each training sample. And in the autoencoder case, we saw that these training examples were stored as attractors. In the sequence encoder case, we stored sequences as discrete limit cycles. And then, of course, now the sequences are easier to store and recover than individual examples. And so this, I think, was a nice theory that, you know, these models can actually do something different than learn the identity function, even when you have a lot of data. And so maybe they're actually useful. And especially based on double descent, we would, I mean, maybe hopefully see that they are better at some of these tasks. And so that's kind of the next question for the second part of the talk, which is how useful are these models in practice? And so what I wanted to go over next is sort of applications that we've seen of over-parameterized models in genomics. And in particular, I'll start off with autoencoders for this problem of drug repurposing. But we'll show that all of these infinite width networks as well are very useful for drug repurposing and virtual drug screening, which I'll explain what these tasks are in a minute as well. I see that I'm not able to annotate, but I think it should be okay. So just as an example of what is drug repurposing, we were doing this actually during the start of the pandemic, when drug repurposing was a very important problem, which is if a new disease comes out and you want to try to treat it, of course, there's a huge approval process. You have to come up with the treatment and then go through FDA approval, and this is very painful. But on the other hand, maybe you already have some drugs that are useful for treating this disease, and they've already undergone FDA approval. And you want to see maybe some of these are actually useful for treating some of these diseases like COVID. And I think a good example was hydroxychloroquine that you know, Trump was spouting on about, that you could repurpose this and get it working. But essentially what we can do is, with the rise of these large genomics data sets, there's a systematic way to actually do drug repurposing that's pretty simple. And so one example I wanted to point out is this CMAP, or connectivity map data set from the Broad. And what they do is they take a bunch of different cell types and a bunch of different perturbations, and you can think about these just generally as drugs. And so they hit these different cell types with these drugs, and they take a measurement, which is called gene expression vectors. And you can just think about these for every cell type and drug combination. you have this measurement which tells you how that drug affected the cell type, and this measurement is usually like a 1,000-dimensional vector. So for every cell type and drug combination, I basically have one point that's a 1,000-dimensional vector. And so the idea is that you can, of course, measure the same vector for a cell type that is diseased. So maybe your cell type is infected with coronavirus. So in that case, a way to actually come up with drugs that are useful for treating coronavirus is you take these points in red, which are my cell type that has been hit with coronavirus, and then I look at this direction to the blue points, which are my control cell states, and I actually see, if I look at the direction that the drug had on these 1,000-dimensional vectors, which of these 1,000-dimensional vector directions correlates with this one taking me from the red points to the blue points. And so the ones that are most correlated would typically be thought of as a potential candidate for treatment. And so this is typically one approach people actually use on the raw 1,000-dimensional vectors. But of course, there's many problems with this. And one of the main problems in this whole field is that these measurements, like 70 cell types and 20,000 perturbations, this is a lot of work to do every single combination. And so typically you have a very sparse data set. And so one application of autoencoders is in the specific case where you want to impute the effect of a drug on a new cell type when you maybe had its effect already on a specific cell type. And so the idea is somehow, can you translate that effect and understand how this drug is going to impact a new cell type automatically for free? And one property that we found of over-parameterized autoencoders that actually lets you do something like this is that they align interventional effects of drugs in this latent space. And what I mean by this is if I look at, let's say I look at many normal cell types, I'm going to call this this cell type in green here. And I encode it with an autoencoder. I think of these as 1,000-dimensional vectors. I feed them into the autoencoder. Then I just look at this direction in latent space that a drug has on my embedding. So here, these points as crosses, I can decode them, of course, and get back a representation of my gene expression that corresponds to a treated cell type. But the idea is if I have another cell type, it's normal cell type 2 in blue, if the drug A affects both of these green and blue cell types in the same direction in latent space, I can just apply that vector and then decode and impute the effect of drug A on this cell type 2. And the nice part is that autoencoders actually give you this alignment, which is a bit remarkable. And this is not just our work. I think a lot of prior work by Fabian Theis's group has actually shown this as well. I think it's just interesting that you can get this effect from a simple over-parameterized autoencoder. And I'll show this quantitatively, what I mean by this, in a minute on that specific data set. But I did want to mention another benefit to over-parameterized autoencoders is actually just the reconstruction loss. So if I look at these essentially 1,000-dimensional vectors of gene expression value, if I just do the simplest thing of one hidden layer autoencoder, I just use all my default parameters that anyone would do in practice. I just train this model, and I compare it to what I would do with a standard under-parameterized autoencoder. You can see here, like, your training loss is an order of magnitude less. Your test loss is actually several order magnitudes less than what you would get from an under-parameterized autoencoder. So clearly, like, your reconstructions are better. But it has not learned the identity function. It's actually learned this function that's somehow aligned interventional effects. And I think this is the exciting thing, that there does seem to be a kind of universal benefit in some way from just increasing the size of this latent space. And quantitatively, what this looks like, this alignment effect, it looks something like this. So if you take two cell types, this A549 cell type and this MCF7, these are two cancer cell types. What I'm plotting here on the top axis is I just look at the correlation, or this cosine similarity, between the effect of some drug I, for A549, and the same drug on MCF7. That's what I'm plotting on the top plot. So the x-axis is what is this alignment between these two cell types for this specific drug in the original space, and the top axis is what is the alignment in the latent space when you use some sort of embedding method. And so here, what's interesting is that if you take your 1,000-dimensional gene expression vectors, you perform PCA on these and you reduce them to 2 principal components, you'll see that you actually get some good alignment in this 2-dimensional space. So like if I take A549 with some drug 1 and MCF7 with drug 2, the angle between those 2 vectors in this 2-dimensional space is very close to 1, or very close to 0. And so that would be nice. That's a good property for actually imputing the effect of a drug on a given cell type. But the issue, of course, is that when you actually do this effect, you need to be able to decode to see what your gene expression values actually look like. And so if you do that with PCA, you, of course, and with 2 PCs, you've lost like almost all this information. It's pretty bad. You can't actually get this reconstruction to work well. So imputation suffers a lot, even though you have this alignment. On the other hand, if you use an autoencoder that's under-parameterized, so it has this bottleneck, if you again compute the same cosine similarity, you'll see that now you don't get this alignment effect. So things that are partially aligned in this latent space, maybe the cosine similarity is like 0.25, it's really not that much better. It kind of looks like the identity. It's still like 0.25 in the latent space. But the good news about autoencoders is that they give you pretty good reconstruction. So you can, if you were able to shift in this latent space and decode, you could reconstruct the data well. But the best part about over-parameterized autoencoders is that it gives you the best of both worlds. So somehow it gives you this proper latent space, where now if I look at any of these samples in 0.25 in the original space, there's many samples here that are close to 1 in cosine similarity. So their angle between these 2 vectors is close to 0. But the best part is that you get perfect reconstruction. I think on the previous slide, the reconstruction error is like 10 to the -6th. So you can actually use these for imputation. And so in this paper, what we did for drug repurposing for coronavirus, we exactly did this. We did, we were trying to impute with this, it turns out the infected cells that we were looking at were these A549 cells, or something similar to these. But we didn't have all the drugs in our database on this A549 cell. But the thing is that since you have alignment with this MCF7 cell in this latent space, we can just use this example from the previous slide of translating to impute the effect of a missing drug. So this is really nice that you get this property kind of for free. And I mean, I should say, like, I think people have come up with all kinds of tricky variational autoencoders and stuff for this, but I'm not doing any of that in this work. It's truly the simplest thing of like one hidden layer neural network, and I train this thing with that. It's like the simplest thing you could do, and you get this thing. So just to summarize this application, I think this shows that for at least for this drug repurposing, autoencoders have some strong benefit. And just to give an overview of this this portion of the talk, I realize I've saved a lot of time for questions as well, but essentially what I wanted to convey from this, the first part, we showed that over-parameterized autoencoders have this interesting inductive bias, so they bias towards solutions that are contractive around the training data. Here's more of a practical finding that not only is this like inductive bias characterized, but these models are actually good in practice. They produce low test error reconstructions. And in this particular case, they can actually align interventional effects, which is a really nice property for anyone who's doing genomics, I think, and even genetics as well. This is a key question of how do you align these directions. And so just in terms of resources, I have some future work that I wanted to discuss in the next few slides, but this is basically what I wanted to convey about this over-parameterized autoencoder work. So I have some links to the 2 papers. The theory paper is the first one linked here, and then we have a general framework for drug repurposing, of which over-parameterized autoencoders is one part. And of course, in these works, I have to thank many of our collaborators. This definitely, especially the second one, could not have been done without many of the biologists and computational biologists in our group. And so, thanks to my many collaborators here, and especially Misha and Caroline for supporting me in all of this. I did want to mention one recent work, actually, that followed up on this drug repurposing component. And I think it's a kind of a nice extension for people here who are interested in infinite width networks and how you can kind of merge these with these over-parameterized autoencoders. But thus far, what we talked about was this idea of drug repurposing, which is maybe I have one cell type where I know basically all the drugs I care about, and I care about understanding the effect of one of those drugs on an unseen cell type. But here there's a much more general question, which is maybe you don't even have the cell type that you're trying to measure a drug on, and you don't have the drug either. So this is just like, you've never seen this drug before on any cell type, but you want to kind of impute its effect still. And this is just to show that this is kind of a realistic setting, especially as people start thinking about knockouts and knockdowns in genetics. There's just too many combinations of these things to be tested in practice. And even the data set that I was showing before, this connectivity map, if I just look at the availability of measurements between cell types and drugs, you'll see that it's actually only very few cell types that have almost all perturbations available. Most of the data is still extremely sparse. And so the idea is, we have to try to do something a little bit better for these cases where we can't just translate the effect of a drug onto a new cell type. We need to actually predict these for unseen combinations. And of course, this is when you see a sparsity pattern like this is of course like screaming some sort of matrix completion problem. And in fact, that's exactly one way to formulate it. Any of these missing entries, we can just think about their whole 1,000-dimensional vector as a set of missing entries in the space. And we want to impute these missing entries. And so one idea is that we can't actually use over-parameterized autoencoders in the naive way to do this, because we can't actually translate the effect of a drug that we've never seen before. So we have to do something better, and the thing that we will do better is use some sort of prior for matrix completion. But before I discuss something like that, I just wanted to quickly mention here, if you do treat this as a normal matrix completion problem, there's been many, many approaches to doing this. I think the most popular one is trying to get at something that's low rank for a completion. Low rank completion here, I hope it's a bit clear, but it will do terrible for this application. And the reason is that I can just make this matrix low rank by putting zeros in all of these missing columns. Like the entire column is missing. This is not like IID missing entries. This is non-trivial to do here with low rank. There are ways to get around it with some reordering of the tensor that you have here, but it's still a bit tricky. So the simple idea that we had, and I mean it seems very simple in hindsight, but we'll see that it's much more powerful in general, is that whenever you have these missing samples, this is exactly a type of supervised learning problem. And essentially what we want to do is, if we're given some sort of cell type and drug combination, and we want to impute the gene expression values for these, all we need to do is build an embedding for what that cell type, for what that drug is, and we need to build an embedding for what that cell type is. And so if you have that information of cell type and drug embedding, then you can just map to what the gene expression value is, and it's exactly like a supervised learning problem. And I think an even simpler example, if you wanted one, is this case of the Netflix problem, where you have users in movies and you want to predict their rating. If you want to do this for a movie that's never been rated before, you'll have like a whole column of missing entries. So low rank is not going to be good for that. But what you can do is just, if you have features of users and features of movies, you can just concatenate the two and do supervised learning. I think the interesting thing about this, while it seems really simple, is that for those of you who are, again, familiar with this, this inductive bias of deep linear neural networks for matrix factorization, this sort of supervised learning approach is a strict generalization of that framework. And you can capture a lot of those. If you wanted to, you could impose a prior that gives you a low rank reconstruction, although in this case, it wouldn't be good. What you actually want to do is, if you have some knowledge of the metadata that you care about, like what is a drug and how are they similar, and what are cell types and how are they similar, you want to encode that and learn a mapping from that. And so that's exactly where, in this specific case, there's many different modalities capturing drugs and cell types. Cell types, usually you have images of cells. Drugs, you have like graphs representing what the compounds are. Here you can use autoencoders or anything that we've developed so far to build these embeddings. I think the cool part is that we have a recent paper where we did this. Instead of using neural networks or anything like this, you can just use the infinite width limit and use the neural tangent kernel to do this map. And if you actually do that map, you get some quite good results on, like surprisingly good results on this imputation problem. And in fact, we just tried some simple embeddings. I mean, we didn't really have metadata over what the genomics data set gave, so we didn't have images in this case. But we just did some very simple, like one-hot or control sample embeddings for these different groups, and we just see that the NTK, or this infinite width limit, pretty much outperforms a lot of the other methods consistently on this data set. And so I think this kind of over-parameterized approach really does have benefit in a lot of these genomics and even genetics problems. And I think we'll see many more of these to come. But for those of you who are interested in this line of work, I know I didn't speak in too much detail. I focused more on this genomic side, but we have many other applications to image and painting, and image reconstruction, and so on in this archive link, and how you can actually do this with the convolutional networks of infinite width and so on. Okay, I know I went a bit fast, so and it was a lot of material, but please let me know if you have any questions. I think this is essentially what I wanted to cover in this talk. But, yeah.
Thank you, Adit. Very nice talk. Questions? So let me ask you this. So why? What do you think? What do you think when? Like, for example, comparing low rank completion, how would you how would you decide when low rank is good and when this method is good? What's the sort of intuition there?
Yeah, I think that's a great question. I think my gut feel is that I mean, definitely a simple case is that if you have some new sample that you're working with, I guess the best case is like if you have a new movie, never been rated by anyone before. Low rank is kind of hopeless because, I mean, there's nothing that you can do in that case. But if you do have like some amount of sparsity, and I guess this depends on this, a lot of these results by Donahoe and Tanner on like how much data you need to impute this. I think then low rank is worth a try. I also think, like, as a simple example, if you have any sort of metadata of like what your entries are actually, what corresponds to your entry. So if you have like users in movies, if I have metadata for users in movies, for sure I would try to take the embedding from those first and then predict the samples, because that's super easy to do in a supervised learning way. If I didn't have the metadata and it was kind of hopeless, then I think low rank is sort of the best you can do. In this specific case, drug and cell types are kind of a little weird because they don't give you explicit metadata, but just knowing that you're the same cell type or you're using the same drug just via some one-hot embedding, that's totally enough to do well for this problem, because cell types have such a dominant effect in this space and perturbations have very little effect. So I think that's why in biology it does make sense to do something like this, but in these other regimes it's a little tricky. Thank you though. Thanks for the question.
Yeah, thanks. Any other questions? Questions, Adit? Okay, let's thank Adit again.
Thank you. Yeah, thank you for having me.